Okay. So today, we're gonna do a little bit more on phloem translocation and we might start the next topic, but the second part of the hour, I'll stop around probably 12:30 and we will talk about the exam and go through any questions you may have on exam content, okay? So that's the plan. We've been looking at phloem translocation in the vascular tissue. In angiosperms, we're thinking about the sieve tube element and companion cell complex. Here you're seeing the full thing, both what's happening at the source and at the sink. Movement, once you are zoomed into the sieve tube element, it's all due to a pressure gradient. And if you develop the higher pressure at the source with the loading in of the sugars, and then as you're unloading the sugars down, or I always say down because I'm thinking about a potato plant that has a developing tuber. You unload the sugars into that tuber tissue. Well, as you do that, that ultimately lowers your pressure in the sink area. And so you get that gradient. And so the plant will keep sending sugars down to that developing potato until that gradient pretty much dissipates. Like once there's not a strong gradient, then it's like, well, okay, I guess it's done unloading. I don't have a strong gradient to follow anymore. And so then they can send sugars elsewhere. And so the plant is constantly assessing that kind of feedback from the sink tissue, trying to figure out where to send its sugar, how much to send. And a big part of how it's assessing all that is this pressure gradient. Is it a really strong pressure gradient? Is it starting to even out in terms of pressure from the source to sink? And then you can see that the xylem is heavily involved because the water coming from the xylem is feeding into this process to develop a higher pressure at the source and a lower pressure at the sink. So we were talking about this last week and we got into how the plant is doing phloem loading. How does it load its sugars? What pathways does it take to get the sugars into the sieve tube element? But we need to also talk about what's happening at the other part of it, at the sink. And so with phloem unloading, that's a little murkier. They have, because there's so many different sinks that you can think of for the plant. And so they've looked at like developing seeds or developing tubers or root tissue. They've looked at a bunch of different things, and I'm going to greatly simplify because it's so varied on how plants do this. So we're going to say a few things and call it good. Lots of sinks out there, the different sink tissue. Basically, remember, sink tissue is anything that can't make enough of its own photosynthesis, the sugars, or it doesn't do any photosynthesis and definitely needs some sugar from source tissue. So there's lots of different methods of how they unload the sugar out of the sieve tube element into the sink tissue. They do witness symplast, that's the plasmodesmata, the little internal tunnels from cell to cell. They probably the best example for that they have the most data is root tips. Root tips are continually growing throughout the life of the plant. And so they've been very interested in that. Seed development is another huge thing. How does the sugar get to the developing seed? And they see a lot of apoplast movement. So that's the cell wall. And it depends on the plant. Sometimes it's just straight ahead sucrose gets delivered into the seed. Sometimes they notice that sucrose gets hydrolyzed to its two monosaccharides, the glucose and fructose, and then that gets imported into the sink tissue. And then sometimes they do see energies not needed to get this to happen. So primary active transport, secondary active transport. That's the idea that you do need to expend ATP, get a proton gradient, and then use that proton gradient to drag sucrose or glucose or fructose into the seed tissue. So the seed tissue is pretty nutrient dense. So kind of makes sense that they have to kind of push the sugar up a very high concentration gradient into the seed tissue. But that's pretty much what we're going to call it in terms of what I want you to know for phloem unloading. This is just a diagram where they've come up with hypotheses on, how does this happen? So here they're showing some apoplastic movement where they're saying it has to cross the cell wall area here. Here they're showing Symplast through the plasmodesmata, and then it might, do both, like switches over from Symplast to apoplast and vice versa. Lots of different ways that they have witnessed it, mainly because there's so many different tissues in the plant body that are defined as sync tissue. So it gets very complex very quickly. So I'm just gonna mention that's pretty much it. But I do want to think about sync tissue a little bit more in the sense that, when the sync tissue is getting all of this lovely sugar, what is it doing with that? And it's similar to what we think about, like with the leaves doing photosynthesis. They keep some of that sugar for themselves before they send off some of it elsewhere. And basically, they are both using it for respiration, which is just trying to get at the idea that they're using sugar, like they're running glycolysis and electron transport chain in their mitochondria, and they're doing all of that in order to get ATP. So you're doing that right now in your body as well. They need it to maintain their cells. Maybe there's damage. They need to repair. So they'll need some carbon for that. They need it to grow the cells and tissues. And then if they have some extra, they can store it. And so some of the cells, and it's usually pretty good with roots. Roots are pretty well adapted to take extra sugars they're getting sent and storing it. And if you look at root tissue under the microscope, you tend to see that they've stored up. Most root tissues will store it in the form of starch. So if they're going to store extra sucrose, they don't keep it as sucrose, they'll convert it to starch. Not all plants do this, but the vast majority of important plants that have been studied do this. So we store up in the form of glycogen. Plants tend to do starch. So it's similar to what we think about what the source is doing, except that sources tend not to store huge amounts. They'll store a little bit, but not a lot. Whereas sync tissue tends to store up more. And then sync tissue can shift. So it doesn't always have to be sync tissue. There are parts of the body where that part of the body starts out as sync tissue, but then slowly over time, as it grows and matures and gets up to its full potential, then it starts becoming source. It'll be source tissue. So it really does depend on the physiological state of the sync tissue and its developmental state. So if you can imagine a baby leaf just starting to form and bud out, and you get this little tiny baby leaf, it's a sync initially because it's small, it's like a baby. It can't get all of its needs met all by itself. So it's sync tissue. So here's an image trying to get across that, where you have a baby leaf on figure A, and all that black is showing that it's imported sugar the CO2 has been labeled. And so they are showing that it's taking all of the leaf tissue is sync tissue. It's like, yep, I need all of that sugar from the source, which is most likely a mature leaf somewhere nearby. Then you can see as you progress to figure B, C, and D, that the leaf gets lighter and lighter and lighter. And so what's that showing is it's not getting all that sugar delivered to it anymore. It's starting to transition from sync tissue to source tissue. And so by the time you look at figure D, now that leaf is bigger, it's mature, it's got everything up and running. And by and large, it is now source tissue. It's shifted. And so this is constantly happening during the growing season, where you have baby leaves that are pretty dependent on mature leaves around it for sugar pretty heavily. But then as they mature, they'll get actually to become the source tissue. Here's another with fluorescent green. I think they use green fluorescent protein with this. And so they said this leaf up here in the top left is your source leaf. And then here, the smaller leaves, and then they said the one on the lower left, that is starting to become a source leaf. So just a different, I think this is Arabidopsis that they used for this image. So there is that dynamic. You don't always have to be sink tissue, especially when you think about leaves. Then there's two words I want you to know, and unfortunately, out in like the regular world, there's a different kind of definition, so this is tricky, and I just want to have you really pay attention to these two words, because I've seen students mix them up on the exam, and I understand why, because out in the real world, you might have learned it somewhat differently. Okay, so in plant physiology, in that world, when you say the word allocation, when you say, and PS is photosynthate, so sugar allocation, essentially, sugar allocation, what does that mean? Well, it is the metabolic fate. When you say the word allocation in this class, what you're saying is, what is the metabolic fate of the carbon or the sugar in your source or in your sink tissue? What's the metabolic fate? And basically, when you are a mature leaf, a source leaf, and you're doing photosynthesis, then you have to make the decision how much sugar should go into storage so I can store up a little bit in case I run short someday. How much should I use for respiration and get my ATP? How much should I transport? So you're allocating, like 1/4 of my sugar goes to storage, 1/4 goes to utilization, and that kind of idea. You're allocating where these different aliquots of sugar are going. So it's the metabolic fate of the carbon in either your source leaves, usually it's leaves, or sink tissue too. You still have to think about that, especially in sink tissues. They have to make the decision how much should get stored, especially as right now in the fall, you have a lot of storage going on. And like I said, most of them will store up sucrose as starch, but a few, especially in the source leaves, they'll store it up as starch. And it's actually in the chloroplasts. It's kind of weird. So they will pack a bunch of starch inside their chloroplasts, especially in leaf tissue. And then in sink tissue, like maybe roots, then they tend to pack away the starch in amyloplasts, which are still plastids. They're just not chloroplasts. So this is, whoops. So they can store it. They can use it for respiration to get their ATP. They can transport it. If they're source tissue, they have to decide how much should get transported out of the leaf to the various sinks. So allocation, metabolic fate of the carbon. What should I do with it? The other word to think about is partitioning, sugar partitioning or photosynthate partitioning. What should I? Okay, so this is more like the source leaf now. The source leaf is making all that lovely sugar, and then it's like, okay, now I have to figure out where to send it. So differential part distribution. And so now the leaf is like, okay, how much should go to a baby leaf? How much should go to that apple blossom? How much should go to the developing apple fruit or the potato tuber? And so it is this idea that you're partitioning your sugars into different parts of your body. Okay. What part of your body gets the sugar when you think about partitioning? One thing that's important to realize is that sink tissue they're competitive with each other, in the sense that one of the ways they're competitive is whoever has the strongest pressure gradient between themselves and the source kind of wins the game. And so if you have a sink that has a really strong pull, so to speak, needs lots of sugar, you'll see that it gets a lot of sugars delivered to it. And so they know that, like, how close the source and sink physically are to each other does make a difference. So, the closer they are, they tend to get some nice delivery of sugar. Also, how direct are the vascular connections between the source and the sink? That also makes a difference. And then how quickly are they, when we say strong sink, one of the big things that's factoring into strong sink is how active is it? And two things, how active is it and how big is it? You know, how big is the tissue? So they actually define it as, they say sink strength is equal to the sink size versus I'm sorry, times the sink activity. How active is it? Is it vigorously growing, needs a lot of ATP and carbon? Or is it reaching maturity and mellowing out and slowing down? Sink size, you know, think about a baby, little tiny potato tuber, just starting to develop versus a mature tuber. And so it's both the size and activity that affect the sink strength. And so when we say strong sinks affect the pressure gradient, if you have a sink tissue that's super active, like, oh my gosh, I need so much ATP, I need all this carbon to grow, that is a strong sink. And it actually will cause a really steep gradient because it's unloading the sugars so quickly out of the sieve tube element that you get a really low pressure. And then you have the higher pressure up in the source. And so you have a really steep or strong gradient. And so that will tell that source tissue, oh, I need to really keep focusing on partitioning my sugar to that developing tiny baby tuber. And so here is a mock-up trying to show the difference between allocation and partitioning. So allocation is just, you know, you're zooming into the metabolic fate. You're just thinking about how much should get utilized for, you know, carbon and energy, how much should get stored away, how much should get transported. Whereas partitioning, you're looking at where to send it, what parts of the plant body are getting the different sugars. And they compete with each other. And one way they do that is by how steep can they make their pressure gradient. And so if it's a nice steep gradient, the source knows to keep sending the sugars that way. Does that make sense? All right, so this idea of allocation versus partitioning. And so right now there's a fair bit of people that might have like apple orchards, whether you do it for a livelihood or just have some in your yard, for example. They are messing with partitioning when they're pruning their apple trees, because what they're doing is they're removing some of the sink tissue and leaving others behind, and that will definitely influence the partitioning where the plant will be sending its photosynthate, its sugars. So you can do that with your houseplants too. If you want to create a different growth pattern, for example, on the plant body, your pruning of the plant, one of the things that you're doing is you're affecting the partitioning of the sugar to the rest of the plant body. So when they are working with sucrose, what I want to point out here is just a couple things. I'm not going to get into the detail of everything on here, but basically when you look here, you have your chloroplast. Okay, so what's happening inside the chloroplast? So this is a visual trying to show you that it's in the chloroplast that you get starch being produced. Okay, and they're summarizing the pathway of how to get sucrose converted to starch, but we're not going to get into the details of that. And then this over here, this is the cytoplasm or the cytosol, the other part of the cell. And you can see that there's a choice here. So the plant, as if you imagine plant leaves doing photosynthesis, and they have all this sugar, and they have to decide how to allocate that. And so they say, well, I'm going to put 30% into starch. And so they'll keep it inside the chloroplast where it's getting made through photosynthesis, and they'll stuff it into starch. And then they say, well, the rest of it I'm going to send out. When they do that, they actually take their sucrose out of the chloroplast. I mean, their carbon out of the chloroplast, the building blocks to make sucrose. And so this triose phosphate, that's a building block that they start with in order to end up with the synthesis of sucrose. So when they actually make sucrose, it's not in the chloroplast, it's out in the cytoplasm. And then that sucrose can either get transported or depending on the plant, they might store up some of it or use some of it. Okay, so this is tightly driven, by the way, and I'll get into it more, a little bit more later. But basically this kind of blue ball there, that's a transport protein, and that tightly controls what's happening in the chloroplast versus the cytoplasm in terms of should starch get made or should sucrose get made. And it's also tightly controlled. So it kind of makes sense that the plant has some really good control on whether or not to convert that sugar into starch for long-term storage or keep it as a readily available sucrose, very mobile, easy to cleave. So it's a more ready energy source and it's easy to transport. So one of the big ways that the plant is partitioning is through those pressure gradient changes. It's paying attention to those. The source tissue is like, oh, I got a really strong pressure gradient to this baby tuber. I'm going to send a lot of sucrose there. Oh, but over here, this tuber is really slowed down. It's pretty much done. I'm not going to keep sending sugars that way. And so pressure gradient changes can be a big thing. They've also looked at other stuff though, too. They recognize that's probably not the only way that the plant is getting signals back to the source tissue. And so they think hormones might be involved. They think just the sucrose concentration inside the sieve tube element might be playing a role. They've looked at proteins and messenger RNA pieces, and they've even looked at certain minerals like potassium. So I'm putting question marks because they're still investigating all of this, even phosphate. So they suspect there's other things going on. They highly suspect hormones, but some of these other ideas are still being studied. And again, it kind of makes sense that the plant has more than one mechanism in play to figure out where to send its sugars. Because if you keep getting that wrong, you're not going to develop very well. And it could sink you in terms of it could cause your death if you don't do this the right way. Here's kind of a nice visual with the source leaf on the left. What they're trying to show is the sugars are labeled with that bright yellow. And so they're showing that a lot of the sugar is getting sent for transport. It's getting dumped into the veins. And now it's, into the sieve tube element. It's on the internal highway system through the sieve tube element phloem, and it's getting sent. And then here's a baby leaf. And you can see that it's still a sink tissue. And you can actually see how the sugar is coming out of the veins into the sink tissue, the developing spongy parenchyma and palisade parenchyma. And so it's diffusing out into that. And so it's a dynamic process, especially when you think about baby leaves all the way up to mature leaves. You have this transition going on. And then other parts of the body, a sink tissue will always be a sink tissue, like roots that are buried in soil. Yeah, they're destined to be sink for the rest of their life. So unless you have plants like orchids that have their roots dangling off, a lot of orchids, most orchids grow on trees and then they dangle their roots, and their roots are green. So their roots are doing photosynthesis. So they're a little different. And potatoes are, I tend to focus a lot when I think about phloem translocation because tubers are kind of easy to think about. And just a little plug for potatoes. They're actually way more nutritious than most people think. They make more than French fries, which are delicious, but not terribly healthy. But I wanted to let you know that they are actually incredibly healthy. No fat, low calorie, no sodium or cholesterol. You get a lot of vitamin C. You get more potassium in that than banana, spinach, or broccoli. You get some good B6, and you get some other minerals and nutrients that you need. And it's actually good fiber if you eat the skin, which you should. The skin is actually really good fiber. And it's actually stem tissue you're eating. When you eat a white potato tuber, it's actually stem tissue. And so the skin that you eat is actually bark, which is a little weird. Very thin bark, but it is bark. The little eyes that we look at a potato tuber, those are actually where stems grow out and can make a new plant. So if you have a potato that has an eye on it, if you bury it in the ground and give it some TLC, it will develop a new potato plant. Don't eat green potatoes. Kind of like green eggs and ham. Don't eat green potatoes. Solanine is a chemical that gets produced when the potato tuber is exposed to sunlight and it turns green and it's toxic. So that's why when you buy potatoes, you shouldn't keep them in bags that are transparent and maybe keep them in a dark corner somewhere. Yep.
Hey, you go to like McDonald's and stuff, you could buy something that. Is that what that is? Is it like weird too?
Yeah.
Do you think that like all of our they do, that's probably not so toxic anymore?
Oh, that's a good question. I don't, well, and with adults, you'd probably have to ingest a fair bit of solanine to get sick from that. Infants, on the other hand, I wouldn't feed French fries to, well, I wouldn't. Infants shouldn't really have French fries, but anyways, it's, that makes a difference to your body size. And so adults can probably handle a fair bit, but I've grown a fair bit of potatoes over the year and I've dug up, so fun growing potatoes. If anyone's done that, it's like Christmas because you don't know what you're going to get, until you actually dig it up. And then it's like Christmas time. It's like unwrapping a present. Unfortunately, some of the tubers that I've unearthed can be green, but it's usually the potatoes that are right at the surface of the soil that have been getting exposed to sun. And that's one reason why you might hear people say they mound up the soil around their potato plants, the stem, they kind of mound it up. And that's one reason, is they're trying to minimize sunlight reaching their tubers. So. But anyways, so actually, I have in my notes, solanine is a glycoalkaloid. If anyone is in the chemistry program, the glycoalkaloid. So I don't know if that's susceptible to heat. Like, could you break that down into a less dangerous compound? I'm guessing you could, but I don't know for sure. So maybe the frying of the French fries would take care of it. But I just wanted to put that out there because potatoes are actually way more nutritious than most people think. And the combination of potatoes and beans is actually quite inexpensive. A lot of college students are struggling with finances and being able to pay all they need to pay for rent and food and tuition and all of that. And actually, potatoes and beans are super nutritious and really quite inexpensive. So just don't make French fries all the time and you're good. So I work a lot, and a lot of my research when I was a postdoc was with potatoes. And so have to give a good positive plug for potatoes. Okay. All right. I think we'll stop there. So Friday we'll get into mineral nutrition. We'll start that. And so this slide set on phloem translocation will not be on the exam on Wednesday. So what will be on the exam, we start with plant and cell architecture. So let me pull up that slide set. And let me reshare. Share it this way. Oh, and then I gotta do this. Okay, so plant and cell architecture. Some of this is good for multiple choice questions, knowing basic anatomy, definitions. So we talked about the idea that plants have cell walls, more than one sometimes. And then we got a little bit into their overall body plan. Shoots versus roots and the active areas of growth. And we got into primary and secondary cell walls. Some of the interesting internal properties of the cells. So do you have questions on this content in this slide set?
Maybe more information, like on the exam from the word slides, or is there gonna be a lot from like the pictures of that?
For this slide set, a lot of information found in the pictures is very useful to study, for sure. So yes, I do have some printed text that you're expected to know on here, but and a lot of it backs up what you're looking at in the pictures. So, but like, so for example, slide 14 here, where we're looking at the vacuole. And then slide 13, another image of the plant cell, and I think I pointed out, you know, the difference here, where you have a small vacuole here. And a big vacuole here, and what is the vacuole, what is it doing, and why is it small there and big there, and so there's some things I don't have that in a text slide somewhere, but I still expect you to think through that and understand it. And like the trigger pressure here, you know, why, you know, when you have a mature cell, you have that lovely big vacuole, which is good, because then when it is full of water, it's going to cause some nice structural support. What else? Very possible. And so if you think about some of the ideas, you know, like simplified plant cell maybe, or some of the internal organelles, I might ask you to think about and label. Usually that's a drag and drop. You've done those on EduCat where the words are there and you just drag it into the appropriate spots. That's usually how I set it up if I do a labeling of a figure or picture. Any other questions on this slide set? Good on that. So then water and water potential. I probably won't ask you a ton of questions on like the water molecule itself. That should be something you've already learned in previous classes. But like, you know, these, so like slides 2, 3, 4, 5, that's just to refresh your memory. But like specific heat, thermal conductivity, heat of vaporization, those I might ask you about. All right, so cohesion, tensile strength, adhesion. And then I'm not going to ask you to like write out fixed first law of diffusion from memory. Not gonna ask you to do any calculations. But knowing the basic idea of when you think about that equation, the big picture idea is that delta C over delta X. You're looking at a concentration gradient that is describing what's happening in terms of the rate of movement. And then with bulk flow, it's instead of a concentration gradient, it's a pressure gradient. And then the other big thing with that equation is that the radius is to the fourth power. That's pretty significant. The fact that bulk flow is so sensitive to how big or, the diameter is of xylem or a water pipe or a hose, whatever it is you're looking at. But you won't need a calculator. You won't be doing anything like that. What about concepts of osmosis and water potential? What questions do you have lingering on those topics? So, yeah,
Stomatocrips, is that the same concept as like
yes, it's just taking it to the extreme. So sunken stomata are a little bit more modest. They're just slightly sunken. Stomatal crypts are like a really extreme example.
Oh, that's like. The extreme sense.
Yes, yeah. Yep. The water potential equation, psi sub W is equal to psi sub P plus psi sub S, that you have to memorize. That one you need to know. And then know how to work it in terms of if I give you a scenario and it might be something similar to this, but not necessarily, but you'll need to be able to talk about the water potential equation and be able to like parse out the different things, like what's happening with psi sub P, what's happening with psi sub S, given the scenario I present. So it's a done deal. You're gonna get something on that, on the exam, where you're asked to think about what's happening to each part of this equation, the psi sub P and psi sub S, and then how does that affect the overall water potential? Then how does that affect how water moves? And you need to be able to describe, like, why is that happening? Like, if you just say, well, pressure's gonna go up. Well, how did that happen? You can't just say pressure, psi sub P is gonna go up and leave it at that. I want to know the why behind it. And so that's demonstrating that you understand the concept of how psi sub P and psi sub S is getting affected. Yeah, right. I mean, if I'm asking you why, that's pretty much going to be an essay question. So yeah, chances are when I give you a scenario, it'll be like a story problem and then you have to, oh, that reminds me. So when I write essay questions, it's not one sentence. It's usually several sentences. And then it's, of course, you'll read through it and then you'll start answering. That's another reason why I come here a little early, because I want you to go back over that before you submit it. Because I've had students accidentally forget to answer all of the question. And I know they could have done it if they had just gone back and reread it. And so it really is sad to have to take points off when, if we just had time to go back and reread it. So reread my question. Oh, by the way, I don't need, fancy paragraphs and all of that. You can bullet things out. And like I've had students, they'll read a question on my essay and they'll kind of write the answer to that. And then they'll read the next sentence and they'll skip a space and kind of write the next answer. It doesn't have to be this beautiful paragraph. All right, you can chunk it out. And so that if that helps you make sure that you've answered every part of the question, that's fine. I don't count on spelling either. As long as I can tell what you're saying. And I'm gotten pretty good at that over the years. But again, to emphasize, please, please, please. Use less negative to more negative. Okay. That will solve getting confused. I've seen over the past students, if they use higher or lower, they get confused. So water moves from less negative water potential to more negative. And if you stick with that, you'll be fine. You have any other questions on water potential? No? Well, how about transpiration? So I think so it's plant and cell architecture, water, potential, transpiration. So 3 main topics. Let me share that again because it's telling me it's not. All right. Transpiration. So this is a process. So when you think about plant phys processes, that's generally a good essay question. I may have some multiple choice on here, but then I might ask for like explain the concept more fully. Like, why is this happening? How does this develop? Not just the surface stuff, but getting a little deeper into it. So both what's happening in the soil, what's happening throughout the plant body, certainly what's happening in the leaf tissue. All of that is relevant to understanding how water moves from the soil into the plant and then through the plant and out. Questions on transpiration? Yeah, so this apoplast, transmembrane, symplast, yes, know the difference between them. But that would probably be a good multiple choice, to be honest. Having you pick, or maybe either multiple choice or a word bank where you match it up, like a matching type thing, maybe. I don't know. I haven't finished making it yet, but those 3, that's a pretty, we've talked about this a fair bit. So I want you to know those 3 different movement methods. So when we talked about what's happening in the soil, and also, so we talked about what's happening in the soil. Let me pull this up. So slide 6, where we're looking at this idea that you get this tension developing because of the curved menisci or whatever, however you pronounce that. As the water is receding and clinging to the soil particles, you start to get those curvatures at the air-water interface. And as that gets smaller and smaller, you get higher and higher tension. And so that, oh, it's the next. So that equation Again, I'm not gonna ask you to do anything with this equation, but understanding this equation is that, with your radius, when you have the radius in the denominator like that, and it gets smaller and smaller, you get a stronger tension force. And so understanding this idea is important. I won't ask you to like spit that out to me or do calculations, but the theory behind it is important. And then it's the same kind of basic idea when we think about what's happening in the leaf with these water clinging to maybe it's the spongy parenchyma cells inside the leaf. And again, you still get that curvature at the air-water interface. And so the menisci get smaller and smaller. And so you get a really good tension. So it's not enough if I ask you about it to just say, oh, it's a tension. You have to explain, how does that develop? Okay, this is a senior level class. So knowing the theory behind it is important, not just the end result. Okay. What else? We talked about the process, and then we talked about what could impact how quickly water moves through the plant body and out through the stomata. So we looked at humidity and wind and air temperatures. And then a little bit more fine examination of how does the stomata movement occur to begin, once the sun comes up, how does that all happen? Any questions on any of that? I think if you just think about your sweaty body and use that as an analogy, we certainly are sweating today.
That's the question.
Yeah.
Is it gonna be like from all the sides, like one kind of essay per slide set, or is it like one thing?
It's usually a concept, like, I mean, this is all transpiration, but I might pick some of this. I won't, on one essay question, I wouldn't ask you to do all of this. It might be like, explain transpiration, the process. And then just oh, that reminds me. So as you're taking the exam, if you have questions, please raise your hand. That's why I want to be here when you take the exam. I want to be able to help you make sure you're really answering the question in the way I'm looking for. I've had students not ask questions and they get their score back and they're like, oh, I knew that. I just didn't know that's what you wanted. So if you are writing an essay answer, you can write it out and then call me over and say, is this what you're asking for? And I will tell you, yep. Or think about this a little bit. You need to think about this other concept. What do you think about that? And see if you can put that in. So if you're missing a piece, I'll try to nudge you without giving it away, of course. But don't hesitate to ask for questions on clarification like that as you take the exam, okay? I want you to do well. And if it's so frustrating as, when I'm grading these, it's like, oh, I bet they knew that. And they just didn't, answer it. I just feel so sad. So it's ask while you're with me. And then Duncan's going to be here most likely too. So you can ask him. He'll also be helping me assess that you've submitted everything. So before you leave the exam or the room, you have to show either him or me that you've submitted both versions of the exam, and then you can leave. Okay, so you have to touch base with one of us. So we need to see your laptop that you've submitted both the essay and the multiple choice. But make sure to ask questions. I'll be the one, mostly answering questions that you may have, and then he'll be helping check that you've submitted both. But even on multiple choice, ask questions on that too. Remember, I'm the one making the question, but your brain's different than my brain. If I write something that's confusing to you, I'm not doing it on purpose. I'm not trying to be tricky. Okay, it's just my brain and your brain might not sync up. So just ask and I'll try to rephrase it. And that I've time and time again, I've seen students like totally like, I don't understand, I don't get it. And then I'll rephrase it and they're like, oh, now I know what you mean. All right, so please ask. Anything else we need to think about? Questions? As the exam, so the exam's Wednesday, so I'll be checking my email into the evening now. So if you have a question and it's something relatively easy for me to answer via email, you can send me a query at night. I just won't be up probably past 11, but you can send me a question that way and I can answer it that way too. Okay, so I have office hours, but sometimes you don't get to it till in the evening when you're studying, so. All right. We'll see you on Wednesday. I'll come a little early. Around 11:40.